The role of tau and PIN1 in Alzheimer's disease. Alzheimer's disease, AD, is the most common form of dementia, a neurodegenerative disorder that affects more than 35 million people worldwide that results in the gradual, irreversible deterioration of brain tissue and cognitive impairment. AD can be caused by increased age, genetic predisposition, brain injury, or environmental factors. AD can be characterized by two neuropathological hallmarks: neurofibrillary tangles formed by the intracellular aggregation of tau protein, and amyloid plaques formed by the aggregation of extracellular beta-amyloid protein. Tau is a highly soluble, intrinsically disordered microtubule-associated phosphoprotein found in axons of mature neurons that bind to tubulin to stabilize and assemble microtubules, which are important cytoskeletal structures that act as tracks for cellular transport in neurons. 6 different isoforms of tau exist in the human brain as a result of alternative splicing of the MAPT gene, which are distinguished by the number of microtubule binding domains, MBDs, half of which have 3 MBDs and the remaining half have 4 MBDs, which are better at stabilizing microtubules. The most common post-translational modification that regulates tau are glycosylation, which affects tau's folding ability, stability, and solubility, and phosphorylation, which facilitates tau's binding to microtubules. As previously mentioned, phosphorylation of tau is critical for binding to microtubules, but it is also important for the regulation of microtubule-dependent axonal transfer of cargo. There are a total of 85 serine, threonine, and tyrosine phosphorylation sites found in the proline-rich domain of tau. Physiological tau is phosphorylated approximately at 10 out of the 85 potential sites in the normal human adult brain. On the other hand, more than half the phosphorylation sites are phosphorylated in tau of AD-afflicted brains. Hyperphosphorylation results in the natively unfolded tau to assemble into paired helical filaments, PHFs, which aggregate to form NFTs. This impairs microtubule assembly and leads to the collapse of microtubules and induces loss of axonal transport and neuronal apoptosis. The proline-directed kinases GSK3-beta and CDK5 are the key culprits involved in tau hyperphosphorylation. This ultimately contributes to the development or progression of Alzheimer's disease. In the following research article, Nakamura et al. Revealed that PIN1, a unique peptidoprolyl isomerase that isomerizes specific phosphorylated serine or threonine proteins from the cis-to-trans conformation by proline-directed kinases, could be used to reversibly promote the isomerization of pathogenic cis-p-tau phosphorylated by CDK5 at threonine 231, the physiological trans-p-tau, to protect against tau pathology associated with AD. They compared and measured the relative levels of cis and trans-p-tau in transgenic mice overexpressing tau-TG, with double transgenic mice expressing tau-TG plus endogenous PIN1 overexpression and double transgenic mice expressing tau-TG plus PIN1 knockout. Both the Western blot and immunostaining results corroborated that the overexpression of both endogenous PIN1 and tau yielded higher levels of trans-p-tau and lower levels of cis-p-tau, which would suggest that PIN1 can restore hyperphosphorylated pathogenic tau to physiological tau via cis-to-trans isomerization. In contrast, the Western blot and immunostaining featuring the PIN1 knockout with tau overexpression showed a stronger band intensity and elevated fluorescence levels of cis-p-tau when compared to just tau overexpression alone. This would indicate that without PIN1, tau phosphorylation would be unregulated and would lead to more frequent hyperphosphorylation, resulting in the accumulation of pathogenic cis-p-tau. Thus, PIN1 plays a critical role in reversing phosphorylation, preventing hyperphosphorylation, and preventing accumulation of cis-p-tau, which could have potential therapeutic implications in treating Alzheimer's disease.